This genetics examination. Question number 1. Enumerate the modes of gene action and determine the phenotypes and genotypes of the progenies produced in each mode. Enumerate the modes of gene action and determine the phenotypes of progenies produced in each mode. There are 6 modes of gene action such as receive, efficacies. Example, 9 purple, 3 red, 4 white. Number 2, dominant efficacies. Example, 12 white, 3 yellow, and 1 green. 3, dominant and receive. Example, 13 green, 3 purple. Number 4, duplicate receive. Example, 9 purple, 7 white. 5, duplicate dominant. Example, 15 own nice, 1 O less rice, and 6. Phylomeric gene interaction. Example, 9 long, 6 on, 1 O less. Question number 2. Difference dominant from received gene. Dominant traits are always expressed within the connected. It is dominant even if only one copy of dominant trait exists. Received traits are expressed only if both the connected alleles are received. 3 question. A dominant jelly is common crop produce normal pigmentation and receive produce gold pigmentation. Cross between normally pigmented and gold common crop produce all normal pregnancies. What are the parents? What are the genotypes of pregnancies? What genotype should be made to produce all gold? What are genotypes of parents? GG normally pigmented, GG phasenomenic pigmented, GG golden. What are the genotypes of the progenies? Female, gametes. What is genotype should be made on produce on all gold phenotypes? It is the GG genotypes produce all gold pigmentation. Based on the theory discussed on hybrid cross, list all the games produced by cross of two homogeneous guppies with curved spine. Games for G. B. Draw Punnett squared and determine the genotypes and phenotypic ratio are expected from the cross. Female games. How are transparent tailed guppies produced? A transparent female guppy can be produced if the male is transparent and female has at least sense allele of the transparent tailed. Male guppies are has received produced only when female. And number 6 question. Determine the factors of causes variation among organisms. Mutation of change. The sequence genetics in DNA are one of source of genetic variation. Other source is gene flow of the movement of genes between different groups of organisms. Finally, genetic variation can be result of sexual reproduction which leads to creation of new combination of genetics. Number 7 question. Understand and apply the concept in selection in fish stock management. A stock assessment is process of collecting, analysis, and reporting demographic information to determine change in abundance of fishery stock in response to fishing and to the extent possible of predict future trends of stock abundance. Managers use stock assessment as a basic evaluate and specificity and present and variable future condition of fishery. Number 8 question. What is genetic drift and how can it prevented? A genetics drift it can change frequency and existing gene variants in the population due to random change. Genetic drift may cause the variation of disappear completely and therefore reduce genetic variation. It could also cause intensely raise allele to become much more request and even fixed. Genetic drift is technically not variable, however, it can be limited by 2 factors. First, the population in question can be significant large enough to accommodate random factors if breeding, with a large enough breeding population, the offspring of each generation will like resemble the genetic variation in the previous generation. Second, the smaller population can maintain similar genetic makeup. The risk of the species through continuous breeding with individual outside of the smaller population. Thank you.